DNA could arguably be one of the most important substances for the basis of all life forms. Our knowledge of DNA today is based on the discoveries from many different scientists over the past century. DNA is the genetic blueprint cells use to replicate themselves and to make necessary molecules such as proteins for survival. In 1953, James Watson and Francis Crick discovered the structure of DNA based on a photo of DNA taken by Rosalind Franklin called Photograph 51, which slowed the double helical structure of DNA. Around this time period, Erwin Chargaff discovered that the A nucleotide binds to the T and the C nucleotide binds with the G. Later on, it was discovered that the T nucleotide was not present in a single-strand template that leaves the cell, called RNA. Instead, this T nucleotide is replaced with a U nucleotide. Now that we know the structure of DNA and a little bit about RNA, how are proteins made? The process is broken down into two steps: transcription and translation. First, transcription is the process of using our blueprint DNA found in the nucleus to make a readable copy of it, called messenger RNA or mRNA. mRNA exits the nucleus and heads towards the protein builder called a ribosome. Once the mRNA reaches the ribosome, the process of translation or protein production begins. To make a protein, many different amino acids need to be joined together. The ribosomes bring in a different helper molecule known as tRNA to read the message from the mRNA while also carrying the necessary amino acid for the protein. Therefore, in the early 1960s, it was established that information travels from DNA to RNA to protein. In 1964, two scientists, Marshall Warren Nirenberg and Philip Letter, wanted to investigate how exactly the mRNA was able to allow a tRNA to bring in a specific amino acid. Their two main questions were: is each tRNA molecule specific to a certain sequence on the mRNA? And how many nucleotides are needed to bring in one amino acid?
Alright, so Letter, we know that we need an mRNA, a ribosome, and a tRNA to create a protein. But how does the mRNA know what amino acid to bring to the tRNA? I think we should conduct an experiment to find out.
That's a great idea, Nirenberg. I have another question though. What is the minimum number of nucleotides of mRNA needed for tRNA to bind and create an amino acid?
It's a great question. Well, we already know that a long chain of U nucleotides will create a phenylalanine amino acid.
How do we only know that the chain of U nucleotides will lead to the creation of phenylalanine?
Well, we create an experiment. We have a beaker, we have all the translation ingredients. So we have energy, we have a ribosome, we have the mRNA, and then we also have the binding factors. Then we're going to add a radioactive compound called carbon-14. We're going to add that to the phenylalanine. If the sequence of U nucleotides binds to the tRNA on the phenylalanine, then it'll stick to the ribosome and we'll get our answer.
Then we can pass the solution through a nylon filter so that the ribosomes stick to the filter.
Great, let's get started.
First, Nirenberg labeled the tRNA with the phenylalanine amino acid as radioactive. This is symbolized by our experimenter, Nirenberg, placing a radioactive hat on our tRNA molecule. The mRNA, symbolized by the students holding hands, is added to the solution, in this case the room, and the tRNA successfully binds to the mRNA. Then, Letter pours the solution through the nylon filter and finds that only the ribosomes could attach to the filter.
Oh look, when I filter the solution, only the ribosomes that are attached with mRNA and radioactive tRNA with the amino acid phenylalanine remained.
That makes sense. If we only use U nucleotide in the solution, only phenylalanine should be made.
Let's use some controls and see what happens when we add the amino acid that we know should not bind to the long chain of U nucleotides.
Sounds like a good idea. Let's try that.
This time, Nirenberg attached a radioactive label, as symbolized by the hat, on the amino acid lysine, knowing that it would not bind to the U nucleotides. Once again, Nirenberg creates a radioactive tRNA, but this time it contains the amino acid lysine. This is illustrated by our experimenter placing the radioactive hat on our tRNA molecule. The tRNA containing lysine cannot bind to the U sequence. Then Letter pours the solution through the nylon filter. Nirenberg and Letter examined the filter and found what was expected. The lysine did not appear. This showed that the U sequence could only bind to the phenylalanine and not any other amino acid. Just to be sure, Nirenberg and Letter also tested another amino acid, which was not phenylalanine, and confirmed again that nothing stuck to the filter. Therefore, they were able to confirm the specificity of the phenylalanine in recognizing only the U nucleotides.
Alright, so we know that only long chains of U nucleotides will create the phenylalanine amino acid. But exactly how many nucleotides will create that specific amino acid?
Well then let's create different lengths of mRNAs and repeat our experiment to see if they bind to the tRNA.
Exactly. I think we should start with 2 and then work our way down and see what happens.
I agree, let's go for it.
Alright.
As before, the experimenter labels the tRNA attached to the phenylalanine with radioactivity as symbolized with the hat. The experiment is repeated. This time, the tRNA is not able to bind to the mRNA, which is only composed of 2 nucleotides. As you can see by the actors, the tRNA is not able to bind to the 2 nucleotides in the ribosome, symbolized by the hug, so the protein complex between all the molecules was not formed. Then Nirenberg pours the solution through the nylon filter. The 2 experimenters examine the filter and do not see any of the phenylalanine, which means that the 2 nucleotides was not enough for the tRNA to successfully recognize the mRNA. They decide to try a sequence of 3 U nucleotides next.
If 2 didn't work, then we should try 3 U's.
The experiment is repeated. This time, 3 actors acting as 3 U nucleotides in the mRNA enter the solution. The radioactive tRNA is able to bind to the mRNA, which is composed of 3 nucleotides. This forms a complex with the ribosome, tRNA, mRNA, and the amino acid phenylalanine that won't be able to pass through the filter. Once again, Nirenberg pours the solution through the nylon filter. Nirenberg and Leder examine the filter and see the radioactively charged phenylalanine in the filter. Since the 3 U nucleotides were successful in binding to the tRNA, Nirenberg and Leder reason that it must take 3 nucleotides to code for a specific amino acid.
That's great. We see phenylalanine. So now we know that it takes 3 nucleotides to create a specific amino acid. And we're going to call those a codon.
We can repeat this experiment using different combinations of codons to find out which 20 amino acids are paired with which codon.
Exactly. We just cracked the genetic code.
This same process was repeated with A nucleotide sequences and C nucleotide sequences to figure out the specificity of the codon to the amino acid. Through the repetition of this process, eventually 61 out of the 64 codons were found to be associated with a specific amino acid. Because of this experiment, Nirenberg and Leder determined that a sequence of 3 nucleotides, or better known as a codon, were able to bind to a specific tRNA carrying a specific amino acid. This allowed us to determine the genetic code that we now use today by repeating their method. This experiment led to a greater understanding of how mRNA directed translation. It was gifted the Nobel Prize in Physiology or Medicine in 1968. As a result of this, scientists are now able to assemble the strands of both DNA and RNA together to produce selected proteins. Also, this experiment is very useful in pharmaceuticals.